While we are all making the effort of social distancing and wearing masks, the real solution to the COVID-19 crisis lies in the creation of a vaccine. But what exactly is a vaccine, and how is one made? Traditionally, to make a vaccine, virus particles are inactivated or killed by chemical alterations. The vaccine, containing inactivated virus particles, is injected into the human body, eliciting an immune system response that produces antibodies or proteins that mark these foreign particles for destruction. But these traditional methods of developing vaccines can take up to 10 years, partly because growing and inactivating viruses on a large scale can take quite a bit of time, and it's painfully clear we don't have that kind of time. So, as we speak, companies are racing against the clock to develop vaccines for SARS-CoV-2, the virus that causes COVID-19. One such approach, currently under development by a company called Moderna Therapeutics, is showing great promise. But before we can discuss Moderna's revolutionary approach, we have to first understand how the SARS-CoV-2 virus impacts our body. SARS-CoV-2, a member of the coronavirus family, attacks lung cells, replicating within them. On the surface of each SARS-CoV-2 virus are spike proteins that bind to an enzyme called ACE2, prevalent on the membranes of lung cells. When these spike proteins bind to ACE2, the virus fuses to the lung cell membrane and releases its genetic material RNA. This RNA contains instructions to create new viral proteins. The host cell reads these instructions to create and assemble new viruses, and when these new viruses are expelled from the cell, the lung cell is thus destroyed. Realize that this destruction could have been prevented if the body had recognized the spike proteins and then quickly deployed antibodies to mark them for elimination. What Moderna is doing is synthesizing the part of the mRNA that encodes for the viral spike proteins and then injects it into the body as a vaccine. Once the mRNA enters the lung cell, the ribosomes in the cell read the viral mRNA, which then churn out copies of the viral spike proteins. Moderna's hypothesis is that the human immune system will recognize these spike proteins just as it would entire inactivated viruses. So later, when the actual virus does enter the body, the immune system will respond quickly because the relevant antibodies have already been developed. Because Moderna scientists have access to SARS-CoV-2's genome sequence in a public online database, they can isolate and synthesize the part of the mRNA that encodes for the viral spike proteins. mRNA can also be mass-produced much more quickly than entire inactivated viruses, so mRNA vaccines have the potential to cut down on vaccine development time significantly. While mRNA vaccines are still experimental and their stability and their effectiveness are still both being tested, one thing's for certain: progress made in developing effective vaccines more quickly will be crucial for battling virus outbreaks in the future.